Okay, well, thank you very much for inviting me to talk today. I'm Siobhan and I'm at the University of Manchester. And my talk is a little bit about the drug screen that we've been doing in our spontaneous model of brain hemorrhage in zebrafish larvae and our identification of ACE inhibitors as a potential repurposable therapeutic for ICH patients. So at the University of Manchester, we use a zebrafish larval model, and this is caused by a genetic mutation, and it triggers intracerebral hemorrhage at 48 hours post-fertilization. And we can visualize this non-invasively through light microscopy and through fluorescence in our transgenic lines. And we use zebrafish because they develop very quickly. They have an innate high fecundity, and their external fertilization of eggs makes them really amenable to genetic modification and drug treatment. And in the UK, prior to 5 days post-fertilization, they're an unprotected species, which means that they offer a really viable partial replacement model, an alternative to the rodents. So we've previously characterized the pathology associated with intracerebral hemorrhage in this model to draw comparisons with what we see in the human condition and with other preclinical models. And we've noticed that there is a consistent and reliable cluster of these dying cells associated with intracerebral hemorrhage in the brain at 72 hours post-fertilization. And we can quantify this using our transgenic lines and the fluorescence for dying cells and the Nexin 5 expression. So this led to the development of a drug screen, a manual drug screen in which we screen for this cluster of dying cells associated with hemorrhage at 24 hours. And then we've gone on to further characterize any neuroprotection with our functional assays and determine if we can find any novel therapeutics for intracerebral hemorrhage patients. From the 2,000 compounds screened in the Spectrum library, we identified 150 drugs that were positive for ameliorating brain damage after hemorrhage. And to cancel this list down, we used the PREDICT and by random walk databases of drug disease interaction predictions based on cardiovascular interaction to limit this list to 6. And out of this 6 drugs, 2 were ACE inhibitors. So we decided to proceed with these due to clinical interest. So in larger sample size numbers, we proved that ACE inhibitors do protect against brain cell death. We quantified this in 30 animals. We also proceeded to note that ACE inhibitors restore a locomotor deficit that we can assay using our spontaneous swimming assay. And that this also offered a genetic neuroprotection mechanism. So we noticed that there was an upregulation of a neuronal marker, ELEV-like-3 in the fish, and a proliferation marker, PCNA, more so with quinapril than ramipril. And in order to determine some more information about the neuroprotective mechanism occurring with ACE inhibitor treatment, we've recently completed a proteomics assay with the protein lysates from heads from these treated fish, and noticed that this potential neuroprotective mechanism could be offered through protection of the blood-brain barrier and ECM receptor interactions, influence over metabolism and lipid processing, and direct implications on cell proliferation. So finally, we wanted to prove the translatability of our idea. So we have retrospectively analysed the INTERACT-2 dataset. So the INTERACT-2 trial was looking at the lowering of blood pressure after intracerebral hemorrhage to look at benefits in 90 days. And when we analysed these patients for those who had administered ACE inhibitors after a hemorrhage, opposed to those who are on prior to the ictus, we noticed that there is a significant increase in the neuroprotection offered from ACE inhibitors, and this translates to a better score of MRS at 90 days. And this was only seen in those patients that were administered ACE inhibitors after a hemorrhage. So future directions, we'll be looking at a little bit more about the neuroprotective mechanisms that are offered through ACE inhibitor treatment, and to determine a mechanism of action, whether this is indirectly through the influence of blood pressure or perhaps a novel direct inhibition of ACE. And we're also interested in determining the importance of timing with drug administration in order to translate this clinically. And I'd like to thank my colleagues at the University of Manchester, our collaborators at the George Institute and the Sheffield Institute, University of Sheffield, and for the audience for listening. Thank you.
Thank you very much, Siobhan. That was really interesting data, and nice to see you sort of following it back into man as well. We're looking at the drug trial.